Is everybody excited? Because I am. I mean, when I get to see the progress that you have made as a Holstein breed, and John, my hat's off to you and your team. I was out to Holstein in Battleboro in the most beautiful part of the year, I believe, early fall, and got to see a preview of Inlight, and I was just astonished. And I can assure you, come Wednesday, when I speak to the Angus Association Board of Directors, they're going to be hearing about Inlight, because that is a phenomenal tool, and you're all very lucky to have it. And I can't imagine what things are going to look like in another 3 to 5 years, John, as we add more technologies, more capabilities to this fascinating field. Okay, here we are. Well, they asked me to come and speak with you a little bit, and I'm not going to talk to you about all the nuances of imputations and all the things that go into the mapping process, but I want to explain to you a little bit today about the history and how we got to where we are. I'm a bit of a technical junkie, as John likes to describe me a little bit, but I've been involved in genomics for now 25 years and started out, I think it's important that you know where I come from. This morning I got up at 4 o'clock and I did chores just like many of you and drove from the deep south of Wisconsin and near Madison up here. And my history takes me through the Meat Animal Research Center in Clay Center, Nebraska, as the author of the first genetic linkage map for cattle. That's the first one that was published. But you all should be very happy to know that really the first map was a Holstein map. And that map was by Michel Georges out of Belgium. How many of you have ever heard of Michel Georges and Genmark? Actually, I have to attribute to the fact that Michel put together the first map, which was then purchased in its entirety, and the company was sold to a company here in Wisconsin called ABS, American Breeders Services. Now, during that time, I was involved at the Meat Animal Research Center doing a genetic linkage map hired from the Ohio State University. You might remember that school if you're from Wisconsin. I'll let that settle in. No, it was a great day if you're a Buckeye. But anyway, that's where I was born and raised. And anyway, at the USDA, we basically developed the genetic linkage map, and we beat the Australians to getting it published. And we used antiquated markers called microsatellites. How many of you have ever heard of microsatellites? Well, those were some of the first robust markers that we were able to develop what we called a linkage map. Because when I started mapping in my PhD program at Ohio State, we had RFLPs, which is even older technology. And when I went to Jim Womack's lab at Texas A&M, there was exactly less than 100 spots on the bovine map that anybody knew anything about. And most of the chromosomes, we didn't even know where they were. We didn't know what was on them. So I mapped a gene called insulin-like growth factor 1, IGF-1. If you've ever heard of that, is kind of an important gene involved in various growth-related traits. But my pathway from Meat Animal Research Center then took me to ABS, where I was hired in to actually combine Michel's work from Genmark with the maps that we had published in 1994 at the USDA and develop the first system for selection in Holstein cattle, and worked with Dave Thorbon at the time down at ABS and his sire analyst team, and we went out and developed a system for selecting bulls within full SIB groups using microsatellite technology. And then I became a director of research and became involved with cloning. How many of you have ever seen this picture of clone cows? These are all identical out of a certain cell line. And then this is a cow. This particular picture, has anybody noticed any of the similarities there? Those are two clones, and the calves in front of them are clones, and those cows carried themselves. And we basically developed that technology at ABS while I was working as a co-founder of a company called Infogen. Some of you may have remembered that. And so that's how I came by Wisconsin, and I've been here for now 20 years. And although I've taken other steps since the Infogen days, I came a-knocking about 2 years ago. Illumina wanted me to come back into the field of genomics and help them develop some strategies for implementation of genomics worldwide. And my particular territory is North, South, Central America. And so I joined the team, and I want to tell you a little bit about the intel inside behind all this genomics that you're hearing about. And that's the way we like to refer to it. Illumina is a worldwide company. We're now up to about 3,600 employees with over 1,200 R&D staff and 400 or so support staff. And we have a large patent portfolio centered around all kinds of technologies, including mostly sequencing and genotyping technologies. Our revenues in 2014 were $1.9 billion. And you can find us on the NASDAQ under the call symbols ILMN. As of noon today, we were trading a little over $202 a share. Our market cap is around $28.6 billion. We service several industries. One of the ones that the one I'm most directly involved is agriculture and companion animals, collectively known as agrigenomics. And we're based in San Diego. Now, I get the luxury of living in Wisconsin, basing out of near Rio, Wisconsin. And the head of the agrigenomic division is about 8.3 miles away from here, residing in River Falls. And basically, we can live wherever we need to, wherever we want to, as long as we can get to an airport. And Delta Airlines is a very good friend of mine. So our products are basically, we are really focused in the company on end-to-end solutions. And what does that mean? Well, basically, we're looking at developing all the consumables, the instrumentation, and the software analytics, and we're now combining that with our base space technology, which is the cloud. And basically being able to put all these things together in end-to-end solution with a complete workflow for genetic analysis from sequencing to genotyping. And as we progress in this field, it's becoming more and more important that we have the capabilities to move large amounts of data around the globe, because there's no one particular site that has all the skill sets necessary sometimes to do particular things. But we can make databases available in the cloud, transport them to anywhere that there's somebody who can manipulate data, for instance, or analyze a set of data and provide data back. And I think there's some exciting things that we'll talk about at the end of my talk coming that'll result out of base space. As I said, our platforms are very, we have a full platform of sequencing. We have a desktop sequencer that prices in at very, very reasonable, about $100,000, all the way up to a $10 million system that allows the sequencing of a genome at roughly $1,000, okay? When they first did Dominant, which was the first bovine sequence, how many of you heard about Dominant? Dominant was the one we based everything on in the cattle world. Dominant cost $50 million to sequence. And now we can do that same depth sequence not so many years ago for $1,000. And I can assure you that we're not very far away from reducing that price even more. And it makes sense that we would do that. I can envision someday that it'll even be less than $1,000. We're not to a point where we can do cattle at $1,000. It's being done in the human field today, and there's various sites around the globe that have implemented what we call our HiSeq 10X, which was introduced last year. It's basically 10 sequencers in conglomerate that basically can do that sequencing. But sequencing has moved from a platform, desktop, can sit on this table, and in basically 2 to 3 days, you can get a 10x coverage of a genome and have a full sequence available for analysis. So it's pretty amazing how fast this technology is moving around. Illumina, 90% of the world's sequencing of humans and animal DNA sequences are done on Illumina sequencers. That's how dominant this company is in the sequencing field. And our markets, as I mentioned, incorporate not only population genetics, we're now into the field of forensics in a big way, biopharma, agriculture, we're down to the consumer level in some areas that I'll talk about, from food safety, microbiome work. We're very involved with cancer research, as well as infectious disease, and reproductive health is really exploding, and then genetic health from the standpoint of diabetes and other inherited diseases. So agrigenomics at Illumina, why did we get into it? In 2008, we basically are very driven to unlock the power of the genome to accelerate genetic gain through predictive animal breeding. We are involved in plants all the way to livestock. We are basically looking at improving what we do in production agriculture from the standpoint of human health. So we are driven with human health. The company is very driven on human health, and nutrition and animal agriculture is very important to it. Now, the tools that are driving this paradigm change, really we began in science very observational in our techniques of evaluating traits. And as we've progressed, we've gone through various stages of various ways to look at something and then determine what genetic variation looks like. And we've kind of come to what we call now the flow cell technology. And this is the kind of flow cells that we sequence on. And I think, Lindsay, I've got some handouts we can give, some clamshells. You can all, I don't have very many with me, so there's a clamshell in there that shows a little bit about the history, and also has a chip that you can take home. It's magnetized so you can put it on your refrigerator. And I'm going to explain a little bit more about that. This genotyping technology that we've developed is various places that it's being used. I won't go over all these. Many of you know genomic selection, parentage verification. There's a whole host of other areas where we've basically deployed that. But what I want to talk about is how did we get here? And how did we end up with these tools that are helping us with predictive breeding? Because in the beginning, back in the day, from 1970 to 1990, we basically relied pretty heavy on pedigree information, as many of you know. And we were very, very focused on dissecting the genome of animals to try to find new tools that would aid us in selection accuracy. Oops. So from 1990 to 2000, we call this kind of a discovery period. When I left the field of genomics back in, well, I guess it was the late 1990s for a while, and really focused on cloning and transgenic technology for a while, we'd kind of hit a wall. There was a little period of time there where we had the RFLPs, the microsatellites. We were trying to fill in gaps and kind of get a better coverage of the genome. And we didn't really have the tool, as we kind of found out with microsatellites and RFLPs, to have a fully implemented system for genomic selection. But then in 2002, which is a pivotal year, there was a paper produced out of Australia by Mywuson and Goddard. And they talked about what would work in genomic selection is if we had some kind of a marker that was randomly distributed throughout the genome, where you could basically find a little polymorphism from one base sequence to the next, and you could track bits and pieces of the genome from one end of the chromosome to the other. And that paper laid the foundation and really rejuvenated genomic selection and drove several companies, including Illumina, to developing some technology to make that happen. And from about 2000 to 2010, we had a lot of innovation going on, and that's during that period is when Illumina was born. Now, Illumina was born around sequencing, but the genotyping part became very important to the early days of implementing genomic selection and genomic prediction, not only in animals, but in humans. It all centered around what we call the single nucleotide polymorphisms, or SNPs. How many of you have heard of SNPs? Okay, and you probably already know all about SNPs, right? And SNPs are basically small base changes, just single base changes in certain DNA sequence. How many different organisms do you know that do not have DNA? Anybody? I can't name one. There's some prions that don't have a DNA in them, but most of them all have DNA. And these SNPs are basically single point mutations from an A to a G, or A to a T, a G to a C, or back and forth, that we can basically identify and track. And when we take those in conglomerate across across all sperm cells, for instance, because every sperm cell represents what? A recombination event, right? If you have sperm cells, there's no two alike. And the interesting thing about it, what makes it work in our bull selection, is someone did a calculation that you know that there are over 1 billion possible combinations of a sire's paternal and maternal chromosome. That's the possibility of a sperm cells that each particular animal has. So how are you going to track that? Isn't that important to know if you're going to do genomic selection? So this technology was, when you combine the SNPs, you can basically track where each piece of that chromosome comes from. And on that chip you've got in your packet, there are various wells, okay? And each one of these can do one particular animal. This particular chip, you can do 24 animals on. And I believe the chips you've got in your packet can do 24 animals. Now, the interesting thing here is this flow cell across here has over 54,000 SNPs in it, okay? And this is called nanotechnology, and I'll show you what that looks like here in a second. And what really, in 2008, when we basically brought this out, this SNP chip had over 54,000 SNPs, and we call it the Bovine Snip 50. How many of you heard of that? You probably heard of it because you've all used it if you're in selection. And this was a combination of several scientists coming together and making this particular set of SNPs. And I can only tell you, it would take an entire afternoon to explain the process of designing a SNP chip. But I was visiting with Jerry Taylor, who was one of the principal authors of the first sets of SNPs, and some others not too long ago. And when they put this first SNP panel together and said, okay, we're going to do this, we're going to make these SNPs and put them on this chip, they had no idea if it would work. I mean, it was literally a leap of faith. So there was no prediction that it was going to work. It was just a grand design and a grand hope that it would work, because those SNPs we'd never been there before. And this was the largest SNP chip in the livestock industry, as well as the first. And this is the driving chip of all chips today. So whether you're using Zoetis, or whether you're using GeneSeq or any other company, and whether they're using a custom chip or what they're making, the backbone of all this was the Bovine Snip 50. And all chips very basically can impute to this SNP chip, which means basically that there are SNPs on this chip for each lane that are in common to all those chips. And that's very important as we go through, because that means if I did a cow today, or I did one 5 years ago, and I do one today, I can cross-reference them. And that's important, okay? So genomically, we can tie everything together. And as we go along, we'll always find new SNPs and better information, as I'll show you at the end of my talk. But it's always going to be important to tie it all back together for obvious reasons. But I wanted to point out that the first SNP chip was the Bovine Snip 50. It was an Illumina chip, 54,600 SNPs on it, and it was a lot of people involved in making it. And basically, this is the machine that it's run on. Very nice little machine, can sit right here on this desk. This is kind of the iteration of chips that have developed till we got to this chip right here. This is the 24 sample chip. That's the one we're currently running. We're evaluating that platform for a potential upgrade here shortly. And when you look at each, this is an older chip, but you look at this particular well here. This is a 12 sample chip. In each one of those wells, there's a little dot. And inside of that are other little wells. This is what you call nanotechnology. This is so cool. I remember the first time when I wanted to find some kind of technology when I was working at ABS to help our sire selection team. I went to Cambridge, England, and I found this little guy sitting out on one of these little streets at Cambridge, and he was doing etching on glass slides. And he was actually etching the glass slide and putting animal DNA on it and trying to figure out how to hybridize things on etched glass. And I thought how cool that was. And this was in 96, excuse me, that I found him. Well, the bead technology had not been developed yet. This was the Illumina technology that started it all when they figured out how to do the bead technology. And basically that each bead, and I'm not going to go into all the steps, but this is really cool stuff, is each one of these beads that sits down in this well has an oligo on it, a little short piece of DNA. If you remember that SNP I showed you, okay, so they basically hybridize one of those little chunks of DNA on each one of these beads, and they put it down in there, all right? And it's all fluorescently labeled for a dye, and that dye is excited by certain wavelengths of light, and it allows us to be able to create a pattern design that looks like this, okay? Now this is kind of the steps that it all goes through, and many of the young people that want to go into science in this field will learn how this all works, but this is a combination of polymerase driving the synthesis and attaching this fluorescent bead on there, this fluorescent dye, and under certain wavelengths of light, it illuminates a pattern like this. Now, how would you like to be a graduate student, and your job is to go down through here on about 100 of those chips and mark down exactly what color it is? That would be like running an abacus, right? It would be like, here we have an odometer, and we're trying to figure out all the different combinations. So, but this all can be done under a laser light, and then grabbed by that information, internalized by a computer, and compute exactly what that SNP was, what that particular read was at that particular site. All that information that comes to you as a genotype looks like that, okay? And it looks, I mean, that's what it looks like under a confocal imaging. So it's pretty amazing. But it allows us to do this, create a genomic pedigree. And most of you know what that looks like. But if you imagine what that is you've got sire and dam, and you've got bits and pieces of chromosome. And with that technology, we can track exactly which piece of that chromosome comes on through to the progeny. And we can tie, if we have the phenotypic information, certain trait inheritance. Now you have the luxury, because of the great database you have in Holstein, of just a wonderful phenotypes. And yet you still need more phenotypes for more things. But what it allows us to do, allows you to be able to do, is to tie certain inheritance for every SNP. So every one of those little dots, I'm going to go back to this. in your analysis system has a plus or minus effect on a trait. So to me, it's exciting. And I'll tell you why. Because when I was learning quantitative genetics, and I had a course by a visiting scientist named C.R. Henderson. Anybody ever remember C.R. Henderson, Walt Harvey, guys like that? Okay. Well, they were pretty important for BLUP, best linear unbiased predictors. There's a theory about in quantitative genetics that we kind of tried to lose sight of for a little while, believing that there was a plus and minus, that every trait expressed was a plus and minus of various pieces of inheritance throughout the genome. And for a while, quantitative geneticists always believed that. And then for a while, when the first molecular geneticists came along, which I was in that wave, I didn't necessarily believe it, but I was in that wave, was, oh, wait a minute, there's major QTLs. So there's major genes that'll be important, and we don't need to worry about all that other stuff. That's just noise. Well, we got excited about that because there was a paper that came out in the tomato. Okay, and the tomato said there was 8 genes, and that was it, that were involved with flavor and juiciness and all that, so we just looked at those. So that gave animal people a lot of belief, molecular biologists in the time, that this would be a very easy task. Well, that's what we hoped it would be, but that's not the way it turned out. And when I remember, like I mentioned earlier, when I stepped out of this field for a while, that's the wall we'd hit. That's the wall I hit at ABS. I had the microsatellites. I could track chunks of DNA in the pedigree. I could give Dave and the sire analyst team a leg up on 4 full sibs. Okay, so they'd have 4 bulls standing there. Which one, Mike, do we choose? I come up with a system that turned out to be very similar to what you have here. I'd say I painted the chromosomes, and I said this chunk came from this bull, this chunk came from this cow, and it must be good because this one has more of the right chunks than anybody else. Okay, so we'd pick that one. It turned out that 88% of the time we were right in the selection in full sib groups. But we couldn't put it on a larger scale until this technology. And this technology now, and the way you do your calculations, allow the plus and minus effects of all these different little SNPs to come together in a massive matrix equation in your BLUP analysis, and it pulls it all down into a prediction. And to me, that's amazing, and it takes a lot of computing power to do it. So it was a matter of time to be able to combine, get the right molecular technology to come back together with the statistical technology so we could make these predictions. And we've gone on to use this technology to find some very interesting information that we can really use. For instance, I just wanted to show you this real quick on basically the identification of 4 loci associated with fertility. And all these SNPs on a particular chromosome lined up, and they basically look at threshold values for the SNPs on those chromosomes. And then when they reach a certain threshold value, and what that is, they set a threshold, that's an individual thing. It kind of says, well, every SNP has a plus and minus effect, but you know, that may be just random noise. But those who have a large effect, whether it's positive or negative over this amount, are the ones we're really going to pay attention to. And that's what this dotted line kind of represents. So you could have set it down here. You could even have set it down in here, but you certainly wouldn't set it in here because you'd get every SNP. But you've got some SNPs here now in this little pool up here that are having a plus or minus effect on a fertility trait. And that's how we kind of select the SNPs that we might use for fertility, is we set some kind of a threshold. So here we've got maybe, we could probably find about 20 SNPs that are basically randomly, or not randomly, they're across all these chromosomes. By the way, there's 29 of these things. And we can find that those SNPs and put those into the equation make sure that we're studying those closely to predict fertility. And that's been amazing to be able to dissect traits like that. And there's been a wide adoption of this technology. As you know, you have the HD chips, that's the 50K, we have the 770 chip, and now the Bovine LD, which is basically the low density chip. But the low density was an effort to basically create a more affordable solution so you can do more animals. And that has just exploded in your particular breed and for the dairy industry. And that is really good because that LD chip is imputable back into the 50K and into the high density chip. So we're not losing any information. We're creating more affordability. And we're basically able to get more animals into that database. And why is that important? Because it helps us to get better accuracy, better prediction accuracy. And the more animals we can do, the more phenotypes we have, and the more animals across the population we can do, the better, more precision we have in making predictions. So it's all a very good thing. Based in 2007 is when the 50K came out. Bovine Snip 50 was the first one, and since then there's been an array of SNPs across many species. I just grabbed this slide to show the progression. I didn't bother to put the plants in there for now, although we do have the maize chip in there because that was kind of an important event. But principally, we're basically expanding these SNP chips for all species, and it's just amazing explosion. So why does genomics work so well in dairy cattle? Well, obviously, it's because you all had the foresight to create a great testing system in the DHIA and to record that information, and you've been very diligent about your breed association registry so we can maintain some great parentage identity. And so there's high accuracy in combining phenotypes and genotypes and animals, and it's really made this technology work well. You are the leaders in application of this technology in the bovine world. Now, there are applications in swine and poultry and all various species of poultry that are extremely accurate, but as you know, I think now they say there's only 4 companies that own all the poultry genetics, basically. So they have a very closed in-house. The swine industry is very closed, and they've got this thing down to a fine-tuned science, basically using these SNPs. And in the cattle world, we have a much more dispersed populations, more people involved, but the Holstein is #1. And that's something to be proud of. Closely followed by Jerseys, not closely, but behind a Jersey, and then the Angus cattle. So the biggest thing I see, as you know, is combining genomics with reproductive technologies. And that gets me back to some of the things I did while I was at ABS, being involved in IVF, and some of the things we did there, and single cell analysis, cloning, transgenics, and combining that with genomics. And the biggest problem you had is you used to have to give talks about, for ABS, 4.5 years to basically prove a bull and spend $50,000 getting it done. At the end of the day, 1 in 10 bulls would graduate, so we'd have 9 bulls going to McDonald's while 1 bull was going to the bull stud or going into the catalog. And now, because of the combination of reproductive technologies, as you well know, we've got people pushing the envelope on genetic selection through heifers and young bulls. And basically, I think Kurt Van Tassel showed me an estimate now where we're down around 2.3, 2.4 years on generation interval from a 4.5. That's having it, basically. And that's just amazing. But it is a bit caution. I don't know how many of you have heard of it, but there is a program going called the 1,000 Bull Genomes. How many of you heard of that? Okay. That's a very important project. It's actually more than 1,000 bulls now. It's in its fourth or fifth iteration of results. Basically, these are some of the bulls that are listed. All these bulls were designated by the breed associations, and they are being sequenced. Principally, we're trying to get a better database, because it can be better than what it is today, of the exact genomes of the important cattle species around the globe. And then we can cross-reference across all those breeds. And you'll see the various numbers for the various breeds that are listed, and that has to do a little bit with the what was the perceived value and economic value for these particular breeds going forward. And hopefully, by taking all those sequences and comparing them, and if we have the right phenotypes across breeds, we'll be able to identify breeds that are more heat tolerant, more parasite tolerant, loss of functions for various traits and things like that, and compare them by breed, and be able to isolate certain areas of the chromosomes where certain influences for traits are coming, and be able to use that information in a more composite way, because believe it or not everyone raises registered Holsteins, and not everyone raises registered Angus. Most of the livestock in the world are composites, crossbreds. And the real objective is to be able to use this technology across the entire population from a predictive point of view, from a tracking point of view, and what have you. In order to do that, we need to deep sequence more animals and get more information so we can actually track certain pieces of the chromosome with great accuracy from one breed to another. And this technology allows us to be able to do that. So in the future, this project, you'll be hearing more and more about it as more and more of the iterations of the runs are completing. I think that project is due to wrap up in the next year or so. And Illumina is involved in that. Most of the sequence that's being derived to be used are coming from various researchers around the globe off of Illumina sequencers. So what does it take to pull all this together? And this is the first computer. And this computer filled an entire room, and most of you have a cell phone in your pocket, or you have one close by that are probably 150 times more, maybe even more, capable than this computer. But it takes vision, and this fella here had a vision. And this vision, I believe, is, well, it was a long time ago. It was way back in the 40s. And this fella built it, and he believed that there would be one of these in every room of every house. He wasn't far off. It just don't look like that. So that was his vision, and it was almost, what, 80 years ago now. So that's what it takes, is it takes vision, and it takes somebody that has passion to make a difference. So we have genomic selection. It's going on in various different species. Genetic diagnostics, as you know, has been around for a while. You've been using it for lethal and semi-lethal mutations, coat color, pollness. Parentage is really a big thing. We see parentage as a huge market. GBS, genotype by sequencing, we're actually moving on. We have the chip technology. Chip technology is extremely cost-effective, but there's also ways to target certain areas of the genome for parentage, for diagnostic purposes. So genotype by sequencing will become more and more prevalent, and you'll see a greater accuracy, greater speed. I anticipate with GBS to lead eventually from decentralization from the laboratories to right by what I call shoot side. When somebody at Illumina says, Mike, they hired me because they wanted me to be able to help them say, where do we need to go? We need to go shoot side. You need to be able to do it right beside your cow. Because how many times do you have to make a decision right there? Usually, quite frequently. So I see a day when we'll have a little USB card, much like this little rascal here. and you'll slide it in, and it'll be able to do maybe 5 or 10 animals. You'll take a hair bulb, stick it in one side, push it on into the computer, the little program will come up, you'll hit the button, analyze. And then all that'll do is it'll sequence, okay? All the information, all the whatever's needed, whether it's some oligo, some consumables, the hair bulb, slide in, hit the button, boom, and that sequence will be targeted. It'll probably be some kind of a GBS, and it'll beam up to a platform in the sky, in the iCloud, base space. Base space, you'll say, I want to know what color, I want to know, is it a genetic defect for this, and what was that? It'll say, what's your credit card number, okay? You may already have that done by PayPal. And all of a sudden, a few minutes later, out of the space will come the results, right there. That's where we got to go. Are we there today? No. Will we get there? Yes. Because there's a lot of people out there awful smart. And that's kind of where we need to be in livestock. Because the decisions have to be made pretty quick. We don't usually have time to spend sending samples around, waiting 3 to 4 weeks for the answers to come back. So I see that happening. Parentage is huge because I don't know about you, but when I was at ABS, I know what the parentage problems were with getting bulls in. And I was shocked at that time. And as good as we all tried to do, parentage is an issue. And if we don't have the right parents, you've heard the old adage, garbage in and garbage out. Well, that's what it's all about. You can't get good, accurate EPDs or accurate genetic estimates when you've got the wrong parents identified with the wrong phenotypes. It just won't work. Besides that, parentage is becoming more and more important from an animal food production standpoint, okay? Because I don't believe we're farmers anymore. I think we're food producers, and we're in the food business. So there's a whole lot of things that come with that. A responsibility to know where these animals come from, how they're handled. So if you have a farm, SOPs, standard operating procedures, written, are going to become a very important to you in the future or in your siblings' future. The kids that are going to inherit your farms are going to know all about SOPs because somebody's going to come to your door and say, well, how do you do this? How do you know that? And you have to be able to show them. Important. So another area, disease diagnostics. We're very focused right now on how can we go out to a farm and look at the microbiome or and study the metabolomics of that microbiome on the farm. For instance, how many of you know how many different strains of E. coli you've got harboring on your farms? You don't. So what the possibilities are from a management point of view is to be able to go out and know where the baseline of the organisms are in your food production systems, and then to be able to manage those over time. Did it rise in this quarter? And if it did, which ones rose? Because which ones rose will be important to know because they'll be affecting the health of your animals. And so being able to do that on a real-time basis and on a farm system will become important. Pathogen surveillance, disease diagnostics, microbiome, those are words you're going to hear, metabolomics, more and more. Epigenetics. How many of you have heard of epigenetics? Turns out this is all very, very complicated. The genome is not a simple place. It's affected by a lot of things. It's affected by how old you are, the things you've experienced. It's affected by what you eat, okay, and when you ate it. And it's amazing. I just recently reviewed on Friday a paper that was that showed that there is some issues with the mitochondrial DNA of mice that were fat versus thin mice. And those had inherited differences that affected the expression of the nuclear DNA, okay? Very, very interesting. We never really knew that before. So my question is, if that happens, and the question really becomes, is when we have overconditioned animals, how much have we affected their ability to genetically transmit what they're nuclearly capable of doing? Because we changed the epigenetic signals. And we never realized that that would become an issue. So what is epigenetics? Epigenetics is these little switches, basically. Think of them as methyl switches that say, transcribe here and use this piece of DNA, or no, don't do it here, use this, or it shuts it down, or it adjusts the timing of transcription. So without transcription, you can't make the protein, so you can't produce it, and then you don't have the cellular process that functioning the way you expected it to. So epigenetics really complicates all the genomics that we've been talking about. And more we're going to understand that. And in the future, we'll combine genomics. We'll have that genomic chip, and we'll also have some form of epigenetics on there that'll be able to say, in this period of time, these epigenetic switches need to be just like this. Boom, 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 in order to get this trait to express in a certain way. So that's coming down the road. We're also very close to being able to do single-cell complete genome analysis. And that is very tricky to be able to take a single cell. And somebody here I was visiting with earlier today sells a lot of embryos, asked a very astute question, well, how are you going to get that cell out of there without cracking the zona pellucida? Everybody know what a zona pellucida is? Think of an eggshell, okay? So you've got the shell around the egg. It holds it all together. You got to get through there to get to a cell inside of that embryo that basically is going to give me that genomic information. But there are ways that we'll be able to do that, and they're not too far off. So, wrapping up, in the future, boy meets girl. Your son meets your neighbor's girl, young girl. They talk about their genomes, what the integration sites are for certain viruses and bacteria. They make some notes. Oh, I don't want to, we're interested in that. They also talk about their heifers. You compare their heifers. Think about the conversation they're going to have. It's kind of amazing. I think about these things now as I've got a grandson. And some of the things I've seen in my short life and what they're going to see. It's exponential, what's going to happen. So, in wrapping up, I believe that successful animal production results from the combined application of art, science, and good animal husbandry skills achieved from knowledge, hard work, and experience, and common sense. With that, I'm going to stop.